nucleophilic, let's say, group. Yeah. All right. So one of those nitrogens اللي موجودة في الإيميدازول in histidine, that's the one that gets phosphorylated. But that's very rarely. يعني most of the kinases, they phosphorylate hydroxyls and serine or tyrosine. Okay. Do you know how many kinases we have? Oh, so far, how many kinases were discovered? Yeah, almost 518. It's a large, a huge number. يعني that's my latest, let's say, information. إذا الرقم, let's say, is bigger, I'm not sure. Okay, مش هنختلف عليها. It's definitely more than 500. So it's a huge number, and many of those are actually implicated in cancer. So that is why يعني research is a huge, and still it's, let's say, it's very, very, very active. يعني لو مثلا بتتابعوا ال FDA approval of drugs, most of those that are concerned in treating cancer are actually kinase inhibitors. Okay. What you see in here is similar to the family tree of kinases اللي موجودة عندكم. هي شوية مختلفة, but it's the same. Definitely you don't have to know the exact names of those kinases ولا إيش subfamilies for them. You only need to know إنه those kinases are subclassified into tyrosine or serine threonine. طبعا later on بس نحكي عن specific drugs for specific targets أو specific enzymes you need to memorize those, بس لما نحكي عنهم later. طيب نيجي هلا نحكي عن the structure of a kinase. الكاينيز structure واللي احنا معنيين فيه is composed of a catalytic domain and a regulatory domain. احنا بس رح نحكي عن الcatalytic domain. That domain, okay, which binds to ATP. All right? من اسمه you think a regulatory domain what does what? ومش كل الكاينيزز بيكون مرتبط فيهم irregulatory domain. طبعا لما نحكي domain احنا بنحكي عن protein, okay? Why it's called domain? Because it's that part of this, let's say, cluster of proteins which regulates the function of kinases. يعني نفس الكاينيزز كمان there are, let's say, many pathways via which their action is regulated. Okay? رح نحكي عنهم برضو كمان later on. هلا again, what we will focus on the structure of kinases themselves. واللي mostly they have, let's say, high similarity between them. Okay? يعني hopefully at the end رح توصلوا لمرحلة إنه بس تشوفوا a crystal structure of a kinase تميزوا إنه a kinase, even if that information was not given to you, because somehow they have high similarity in structure in terms of the N loop, إيش mostly فيه ال C loop and so on. Okay. هلا most of the catalytic domain is composed of almost 300 amino acids. It contains, let's say, an area where ATP binds in. Okay, it is composed of, let's say, an N loop, a C loop, and also it has highly conserved amino acids, وهذول اللي احنا رح نحكي عنهم زي اللايسين, الجلوتاميك اسيد اللي موجوده بالسي هيلكس, let's say الجلايسين rich loop, ال DFG motif and so on. Okay, هذول اللي احنا رح نركز عليهم. وهاي رح نحكي عنها later on. يعني هلا مثلا لو كانت ديزاين انه فقط ترتبط محل ما بيرتبط بس ال ATP for sure, لانه هاي الاريا بتكون most of the amino acids فيها conserved. بس those, let's say, inhibitors اللي رح نحكي عنهم اللي هم type 1 و type 2, they also exploit a hydrophobic region that is not occupied by ATP, وهي بتختلف من one enzyme to another. في عندي what we call a gatekeeper residue, هاي برضو كمان بتختلف from one enzyme to another. Another thing in type 2 اللي هم more selective يعتبروا من type 1, they exploit what is known as an allosteric binding site. برضو كمان هاي الاريا تماما بتختلف from one enzyme to another, أو فيها variations كبيرة. Okay, selectivity is a major issue. يعني أي researcher بيشتغل على kinase على سبيل المثال إنه مثلا he or she are targeting a specific kinase. واحدة من الأشياء اللي for instance after discovering potent inhibitors إنهم they have to show إنه this drug or that compound that is potent against that kinase إنه selective. usually بينطلب يعني مثلا لما إحنا بنشتغل على kinases بينطلب منا selectivity profile إنه ينعمل screening على طبعا مش كل الكاينيزز بس at least اللي معروفين انهم structurally related in terms of let's say الامينو اسيد sequences ما بيناتهم من ناحيه ال for instance الهيدروفوبيك باكيت الالوستريك باكيت ايش الامينو اسيدز اللي فيهم. فيعني لو for instance a drug that was found very potent against a certain kinase ومش selective, usually this will not يعني على الأغلب هالدرug ما بيكون very successful. اوكي إذا ما كان very selective. لكن أحيانا أخرى approaches ثانية إنه بالعكس إنه أنا إذا عندي more than one kinase all are implicated in cancer, بالعكس أنا الأفضل إنه يكون يعني theory wise وحتى كمان in practice أو in clinically wise الأفضل إنه يكون let's say عندي a drug that can act on all of those kinases اللي معروف إنهم they actually are upregulated in cancer. فأحيانا بيكون لها let's say إيجابيات وسلبيات حسب إنه هاي الكاينة أو إنه this drug what kinases it targets eventually. Okay. يعني السلكتيفيتي مطلوبة أو for instance non selectivity مطلوبة إذا كان الدرج بيشتغل على kinases that are all let's say implicated in cancer. The image you see in here shows you two different representations of a خلينا نحكي a representative cancer as a kinase structure. This one or that kind of a representation we call it a cartoon like, okay, and this one is a surface representation. Okay, and this one يعني it shows you let's say only the surface of the enzyme. It doesn't show anything inside. Alright, يعني فقط الكافيتيز اللي بتكون ظاهرة للعيان. Alright, هلا all of kinases are composed of what we call the N lobe with C lobe. Okay, I'm sure you remember إنه any polypeptide بغض النظر عن إيش حجمه أو how many amino acids it is composed of, it is always or it always has what we call an N terminus, right, and a C terminus. The naming of those lobes, okay, is because this is the beginning of the polypeptide chain and that's the end of the polypeptide chain. Okay, look at the N terminus. What is it mostly composed of? Exactly, يعني secondary structures of any protein اللي هم البيتا sheets with C helices. If you look at the N lobe, it is mostly composed of beta sheets, okay, and just one helix واللي احنا بنسميها let's say C helix. All right. That C helix has a conserved amino acid رح نحكي عنه بالتفصيل. هلا this one that is highlighted in red is called the glycine rich loop or P loop. What do we mean by يعني when we say glycine rich loop? What do you think this means? Yes, many amino acids that comprise that loop are actually glycine. You will see later إنه مثلا لما إحنا نطلع على الactive والinactive conformations of kinases إنه الglycine rich loop مكانه بيتغير. Okay, يعني in particular لما نشوف مثلا الactive conformation usually ممكن عندي الglycine rich loop. Okay, بيكون after the entrance of the ATP it covers let's say the binding site حتى to stabilize a residence of ATP inside the active site. Okay, هلا what connects the N loop to the C loop, this loop in here is called the hinge. This is very important. The type 1 or type 2 inhibitors راح نشوف إنه a very important let's say part of those inhibitors they actually form interactions مشابهة للأدينين. يعني that hinge region الأمينو acids will backbone of that hinge region they actually form interactions with ال ATP اللي هو natural substrate والinhibitors كمان. بالذات اللي احنا بس رح نحكي عنهم type 1 و type 2. Okay? Activation loop. Okay? رح نحكي later on بس نحكي عن ال activation أو how the enzymes أو how those kinases get activated via phosphorylation of serine, tyrosine, threonine موجودة عندي على ال activation loop وكيف الconformation إنه تماماً بيتغير. And that change in conformation is what allows for الاوبننج، او خلينا نحكي انها بتتسع عندي هاي السبيس ما بين الان لوب والسي لوب، او اللي احنا بنسميها الهنج region where the ATP binds in. This structure that you see here in yellow, this represents ATP. Okay? كل الكاينيزز بيرتبطوا بالATP. أو خلينا نحكي most of them، يعني almost the majority of them they bind to ATP and that's how they phosphorylate their substrate proteins. Here's another image، يعني هي تقريبا شوية هيك more detailed، بس يعني راح أرجع وأعيد almost نفس information. Again, N loop, C loop. Most of what comprises an N loop are beta sheets, except for the C helix. C helix has a very, very important conserved residue that is, let's say, glutamic acid. E stands for glutamic acid. طبعا the number that follows, that's the sequence number. Sequence number بختلف from one kinase to another. لكن دائما أنا بيكون عندي على C helix A glutamate. يعني سوري بهالإيمج حاطين وكأنه في عندي positive charge, but this is not the case. يعني أو ممكن تكون a lone pair و negative charge وحاطين negative charge بينهم. بس for sure يعني this could be the issue, but it's definitely not a positive charge. لأنه you already know إن the glutamic acid is actually a residue that has a carboxylic acid that will be mostly present at physiological pH, protonated أو deprotonated أو ionized, right? So it definitely would have a negative charge. هلا coming down from one of the beta sheets ومش ضروري تعرفوا which one, okay? A lysine residue, K stands for lysine. والlysine هي عبارة عن butyl that has an amino group at the end, a primary amine that will at physiological pH protonated and bearing a positive charge. Those two will form an ionic interaction ما بينهم, or what they form what is called as a salt bridge, okay? And lysine and glutamate, they will anchor the oxygens of the phosphate. وكأنهم راح يثبتوا الفوسفيتس بمكانهم في ATP حتى يسهل على هالكاينيز أو حتى يسهل على السبستريت ال OH اللي فيه تعمل أتاك على التيرمينال فوسفيت ويصير عندي phosphorylation. Okay? طبعا these are not the only interactions, but this is an interaction that is seen in almost all kinases. يعني لما نطلع عليهم دائما look for a lysine, دائما look for an aspartic acid as the glutamic acid, and you will see both of those amino acids اللي موجودين عندي في الN loop. Okay, this is where the ATP binds in. That's the hinge region or the active site. That's the adenosine part. These are the 3 phosphates. This is where the substrate would approach the binding site or the active site of the kinase, where the hydroxyl of the tyrosine, serine, or threonine gets activated by a glutamic, an aspartic acid, where it will attack the phosphate or terminal or phosphorus in terminal phosphate. Okay. هلا this one in here هاي جزء من this is another protein. ممكن يكون another enzyme, specifically ممكن يكون another kinase. Okay, يعني in other words السبستريت اللي راح يصير له phosphorylation. احنا مش راح ندرس الانتراكشنز ما بين هاي السبستريت وما بين الانزيم. احنا بيهمنا اللي هو ال ATP binding to the kinase. Okay. طيب هلا for sure we've already mentioned what we mean or what a DFG motif. هلا لما نحكي motif by the way, what do you think motif means or motif? Yeah, it's a segment of amino acid that is conserved also in kinases. So by the way لما احكي انا conserved عشان بس بدي اتاكد انه واضحه. Yeah, in all of the kinases you will see that بس عشان اكون دقيقه in almost all of the kinases مش كلهم. That motif, this is something, let's say, conserved in all of them. هلا DFG stands for what? D. Yes, aspartic acid. Okay, G أو سوري الأف phenylalanine. At least اللي احنا رح نذكرهم يعني you don't have let's say to memorize all of them all of the 21 amino acids بس at least اللي احنا رح نذكرهم. Okay, والجي سهلة هاي glycine. Okay, this is very important لأنه later on رح تشوفوا إنه أحياناً أنا بميز الأكتيف من الانأكتيف conformation من let's say ال relative positions of the DFG motif. Okay, يعني ممكن واحدة a very typical question in this course إنه مثلاً تنسألوا أو تجيكم image وينحكى لكم for instance that inhibitor does it bind to the DFG in conformation or the DFG out conformation. So you need to know that. Okay, ممكن ينعطى لكم image أو حتى أعطيكم the structure of the drug itself وتميزوا. هلا as I have mentioned where the ATP binds it forms hydrogen bond interactions مع الهند region. يعني هاي الإيمج اللي شايفينها هون وكأني أنا بتطلع على الإنزيم from top. Okay, as if I am looking down the end loop to the active site of the enzyme. All right, this is the backbone of the Hinge region, that loop that connects the N and C loop. Okay, and in that active site there is a place for the adenine. Okay, and the ribose pocket and a phosphate. أنا بيهمني تميزوا إنه الأدينين okay forms hydrogen bond interactions with the backbone of the Hinge region. لأنه type 1 و type 2 inhibitors, those two types of kinase inhibitors, they actually bind أو بيكون في جزء من الدرج بيرتبط محل ما بترتبط الأدينين and it forms hydrogen bond interactions with the kinase. Okay, or with the Hinge region of that kinase. Okay, usually 2 to 3 hydrogen bonds. أحيانا في بعض الدrugs 1 أو 2 أو أحيانا 3 hydrogen bond interactions. يعني رح تشوفوا later إنه أنا إذا بميز مين اللي بيرتبط بالهنج region بميز مين اللي رح يرتبط بالهيدروفوبيك باكيت ومين اللي رح يرتبط بالالوستريك باكيت إذا في وبالتالي بحدد إنه type 1 ولا type 2. هلا على امتداد الهنج region okay there is what we call a gatekeeper residue. Do you know what gatekeeper means? زي كأنه حارس بوابة. Okay? هلا where the ATP binds there is a pocket okay to its left. يعني لو أنا مثلا بدي أتخيل I'm inside the active site of the enzyme وأنا وراي الهنج region. Okay? To your left there is a hydrophobic pocket. Or in other words, a space inside the kinase binding site, which is not occupied by the ATP. ليش بنعتبره إحنا أو بنسميه hydrophobic؟ لأنه تقريباً most of the amino acids that are surrounding that pocket are hydrophobic in nature. So that is why the type 1 or type 2 inhibitors, wherever, let's say, occupy a hydrophobic pocket, usually should be a lipophilic group. You will see that when we look at those inhibitors. هلا at the beginning or at the entrance of that hydrophobic pocket, this is where the gatekeeper residue ends. رح نحكي هلا عن أو shortly عن ال gatekeeper residue, the importance of that gatekeeper and how it plays a role in the selectivity among the kinases. Okay? طيب هلا there is also another pocket in here which we call an allosteric pocket. This is occupied mostly by a type 2 inhibitors. Don't worry about, let's say, those diagrams. أحياناً 2D images يعني بتخلي صعب عليكم إنه أنا كيف أنا بدي أتخيل. فالbest way to understand all of those two images أو 2D images نطلع على crystal structures. And this is what we will do, let's say, shortly. بس يعني at least على أساس تكونوا هيك شوية your eye trained إنه يعني بالامتحان للأسف فيش مجال إنه نفرجيكم let's say 3D images. that is why we will only have those. لكن if you can train yourself to see crystal structures حتى لو بنعطيكم maps of the active site بتصير سهل عليكم تميزوا. اوكي هلا if you look at this one, all right, can you distinguish where the ATP is? Which one? بأي color موجود؟ ايش هو؟ اوكي ماشي. لو مش محطوط عنده ATP كيف بتميزوه؟ يب في عندي ادينين، في عندي رايبوز والثري فوسفيتس اللي موجوده هون. اوكي so that's the ATP binding site. For sure this one would be the N or the C loop. The N, what lies above the ATP, for sure that's the N loop, and here it's the C loop. Okay, recall حكيت لكم انه there is a very important residue in kinases which is lysine, and in the C helix of the N loop there is a glutamic acid, okay, which is shown in here. لاحظوا انهم they are forming ionic interaction بس ببينوا وكأنهم let's say covalently bound together، بس لانه الايمج يعني زاويه الايمج الاكسجين هي يعني behind it، بس بتبين وكأنها جنبها. Those two are actually forming interactions مع الفوسفات. Okay. طيب. Let's look at that crystal structure for real. يعني hopefully أنا حكيت مع مركز التعلم أو جماعة الحاسوب إنه يعني I'll do my best إنه أنزلكم هنا software، و by the way you can download it for free. ممكن تعملوا يعني إذا تعلمتوا عليه، وهو user friendly نوعا ما، ممكن يساعدكم إنه لما تطلعوا على الكريستال structure مثلا you can manipulate it in the way you want. يعني هون شوية أنا شو ممكن أعمل بالكريستال structure شوية limited. أوكي لكن لو أنا فرجيتكم على الستركتشر وحكيت لكم can you confirm that it's a kinase ولا لا؟ Can you do so؟ كيف ميزتوا أو كيف ممكن تميزوا؟ Yes. So you have two loops. One is smaller than the other. Most of what is comprising this part are beta sheets. That's the C helix, okay? And that's the C loop. تمام؟ And that space in between them، أو خليني هيك ألف شوي أفرجيكم إنه what connects the N loop to the C loop، أو أظن الأصح أحكيها loop مش loop، is this, let's say, loop in here that is called the hinge. Or if you would click على ال ATP, all right, you will see إنه that, let me click again. Yeah, it is forming hydrogen bond interactions with that hinge region. Okay. طيب حكيت لكم على امتداد الهنج region، بس خليني احطها بزاويه صح. يكون في عندي residue بنسميها احنا gatekeeper residue. Okay. Look at this one in here. This residue واللي هي if you would click on it, it's leucine. طلعوا وكأنه انا في عندي space ورا هاي الليوسين. Okay. There are, let's say, يعني لو أنا مثلا بدي أحدد surface راح تشوفوا إنه في let's say space behind that residue, okay, that can be occupied by drugs type 1 و type 2, all right. هلا why we call it a gatekeeper residue؟ لأنه its size will govern مين ممكن يدخل مين ما يمكن يدخل. راح تحدد لي size of the hydrophobic group اللي ممكن تدخل لهاي الهيدروفوبيك pocket. Okay. Let me rotate it again. لهالدرجة شوي بس. ممكن بس هلا بحاول فيها، بس للأسف أنا بعمل السيرفيس اللي هي محل الليجند بس مش السيرفيس حوالين الليجند. السوفت وير أنا بدي أنزله على أساس إنه yeah you can do so، راح تبين معانا الهيدروفوبيك باكيت very nicely، الألدوستريك باكيت very nicely. فيعني إن شاء الله إنه يعني by next lecture hopefully، ولو للأسبوع الجاي إن شاء الله راح أنزله وبنشوفهم. اوكي. بنزل لكم اياه، بنزل لكم لينك على الاي ليرنينج. هو باي ذا واي يعني هي سوفت ويرز احنا بنتعامل معاها او بنشتغل عليها عندنا في الكليه، بس طبعا الشغل اللي احنا بنشتغله بيحتاج يعني لايسنس، وبيندفع عليها الافات يعني. بس بنعمل فيها جوبز يعني a lot. بس the nice thing about هاي two companies انهم they offer free academic version، يعني لاي حدا مثلا interested انه يستخدم السوفت وير تبعهم وبس reviewing، يعني you cannot بنشتغلش عليه، يعني ما بتقدر تشتغل عليه شغل الريسيرش، بس انه you can let's say download crystal structures و you can manipulate highly crystal structures. It's really nice. فرح انزل لكم الويب سايت، بس بدكم تعملوا عليه ساين اب، بيودوا لكم let's say ايميل to confirm بالذات انه انتم من انستتيوشن، و you can then download that free let's say version. وان شاء الله اذا كان يعني very soon if I download it in here بفرجيكم كيف احنا we can let's say what to do to manipulate the structure، to zoom in على الليجند، نشوف السيرفيس حواليها، الانتراكشنز بشكل كثير احسن، بيعطيني السيرفيس للبوكيتس بتكون كثير واضحه وكثير نايس. يعني هون للاسف يعني احنا بس شفنا هيك انه في سبيس ورا الجيت كيبر ريزيديو، بس ما بيفرجيني let's say السيرفيس around it. يعني للاسف هون حاولت كثير، بس بالليجند يعني you can let me يعني show you ligand for instance add representation you can do molecular surface، بس بعمل surface حوالين الليجند فقط، يعني بعمل surface حوالين البوكيتس اللي let's say سواء occupied أو non occupied by the ligand. okay. اوكي، بس بدي افرجيكم اللي هم التو امينو اسيدز اللي حكيت لكم conserved in all kinases اللي هم okay this one in here شايفينها this one that's lysine and that's the glutamic acid from أو that is protruding from the C helix. ولاحظوا الانتراكشنز اللي عاملينهم together along with the phosphates of the ATP. Okay, those interactions is what activates the phosphate so that it would be easy for the hydroxyl in the substrate to attack at terminal 1 and gets phosphorylated. Okay. هلا again back to the gatekeeper residue. As I've told you, the gatekeeper residue is a residue that is at the entrance of a hydrophobic pocket اللي بتكون خلينا نحكي على يسار ال ATP binding site. طبعا هاي ال gatekeeper residue okay it doesn't matter when ATP binds. ال ATP does not occupy that hydrophobic pocket. يعني فقط الأدينين بتكون قريبة منه، لكن the rest of the structure بيرتبط بأماكن مختلفة. هلا for instance for those two kinases here the gatekeeper residue is a methionine and this one is what? لا it has a hydroxyl threonine. okay هلا size wise مين أكبر؟ methionine. okay هلا for ATP it doesn't matter whether it's methionine whether it's serine because again this hydrophobic pocket is not occupied by ATP. But when you move on to drugs, when you design a drug where you have a group that should enter the hydrophobic pocket, for instance, for like this one, for this kinase that has a methionine, for this naphthyl to enter the hydrophobic pocket, okay, this big group will not allow a naphthyl to enter. As here, the threonine, لأنها smaller, that naphthyl can enter and form interactions inside the hydrophobic pocket. Okay? بمعنى آخر, this kinase inhibitor will selectively bind to this kinase but not this one because of the gatekeeper residue. This is how the gatekeeper residue determines the selectivity of different kinase inhibitors. طيب هلا الكاينيزز زي other enzymes زي any other protein they can adopt many conformations. يعني بديش أحكي an infinite number of confirmations بس a huge number of confirmations. إحنا فقط interested بالactive والinactive confirmations. Okay? هلا inactive confirmation is that confirmation where the ATP binding site somehow is not accessible. يعني بيكون صعب على ال ATP to enter that binding site. Where the active confirmation بتكون عندي ال ATP طبعا بهاي الايمجز مش كثير واضحة هلا بفرجيكم let's say another image. That ATP site بيكون عندي more accessible for ATP to bind in. الانهبيتورز اللي راح نحكي عنهم واللي هم type 1 و type 2 those inhibitors one favors the active conformation and one is more suitable to bind to the inactive conformation. اوكي هلا انكم تعرفوا شو كل التغييرات اللي بتصير بالكاينيز ما بين الاكتيف والانكتيف كونفورميشن اكيد لا انا فقط معنيين بالدي اف جي موتيف والاكتيفيشن لوب كيف بيكون شكله اوكي هلا there is almost always an equilibrium between the active and inactive conformation. Okay? Mostly بيكون toward the inactive conformation. لكن في عندي many things that may shift the equilibrium toward the active conformation. منهم اللي هي يصير phosphorylational kinase itself. The phosphorylation itself is one way via which the kinases can be activated. يعني they actually can phosphorylate each other and this is how they can get activated. Another method via which they can get activated اللي هي الارتباط allosteric أو signaling molecules زي مثلا الcyclic AMP. يعني الcyclic AMP in some kinases إذا ارتبط فيهم بصير لهم activation. Okay? أحيانا the translocation of the kinase from one site in the cell to another this may drive the activation into the active conformation. اوكي احنا فقط رح نحكي عن انه if for instance phosphorylation occurs for a kinase how that will change the conformation or will stabilize the active conformation. Let's look at this enzyme in its inactive conformation. Okay? وin particular خلينا نركز على what we consider or what we call the activation loop. على الactivation loop في الكاينيزز they have يا إما تيروسين أو أحياناً سيرين أو أحياناً ثريونين. Alright? The one shown in here is what? Tyrosine. Okay? هلا لاحظوا كيف أنا عندي طبعاً for sure this is the N loop, that's the C loop, this is the C helix, that's the binding site of where the ATP will bind in, that's the glycine rich loop. هلا لاحظوا كيف بيكون عندي part of the activation loop وكأنه مسكر هال ATP binding site. When this tyrosine gets phosphorylated, the phosphate group will increase or decrease its water solubility. Will highly increase it because it's an ionizable group, okay? هلا because of that, then it can, let's say, rotate itself towards a solvent, the water. يعني it will have favorable interactions مع the water molecules. That is why it will rotate itself. And when rotating كل شيء مرتبط فيها will also change its conformation. لاحظوا كيف هلا الاكتيفيشن لوب وكأنه شوي بعد عن ال ATP binding site which will make it now accessible for ATP to comes in and binds in into the active site or the binding site of the kinase. Okay? That's how phosphorylation of kinases will shift the equilibrium towards an active conformation. All right? هلا نيجي ل what we call a DFG motif واللي هي احيانا انا ممكن اميز يعني ممكن مثلا اميز انا الاكتيف والانكتيف كونفورميشن مش من الاكتيفيشن لوب ولا من let's say the glycine rich loop من الدي اف جي موتيف itself. هلا الدي اف جي موتيف that is composed of a aspartic acid phenylalanine and let's say glycine that or the rotation of that DFG motif is what is involved أو خلينا نحكي مرتبط ب conformation being active or inactive. هلا let's look at this inactive conformation. This one is here or this one here is the activation loop. Okay? Now, the one that is in yellow represents a DFG. Okay, this is the aspartic acid, and that's the phenylalanine of this motif. Okay, لاحظوا كيف بتكون عندي أنا الأسبارتيك acid وكأنها pointing away from the ATP binding site. That is why we call it DFG out. وطبعا برضو كمان the relative position of the phenylalanine راح يتغير. Okay. Now, if you look at the active conformation that is shown in red, لاحظوا activation loop now, that part that got phosphorylated in case كان activation by a phosphorylation is now, let's say, is trajectorying toward the solvent. Okay. A DFG motif in here, that's the aspartic acid, is pointing where? وكأنها toward the active site أو binding site of ATP. That is why we call it DFGN. وبرضه كمان الفينيلاlanine, the relative position of the phenylalanine بيتغير. This is very important الفينيلاlanine وهلا رح نحكي عنها ليش. Okay. هلا بفرجيكم let's say ال other crystal structures بتكون أوضح. Inhibitors. These are, let's say, images where here that inhibitor that favors binding to the active conformation of what we call a DFGN, that's actually an inhibitor that is classified as type 1. Okay, here this one is an inhibitor that is classified as type 2. Type 2 kinase inhibitors favors an inactive conformation or DFG out conformation. Okay. Here's another also representation for the DFG motif. Don't worry about it. هلا بفرجيكم الكريستال structures إن شاء الله بتكون أوضح بالنسبة لكم. طيب, as I have mentioned, إحنا بهالكورس رح نحكي فقط عن type 1 و type 2. Okay, where type 1 favors binding to the active conformation. طبعا both of them they occupy الأدينين pocket. يعني بيرتبطوا محل ما بيرتبط ال ATP، بس طبعا مش بكل ال positions where ATP binds in. الإشي الكومن بينهم وبين ال ATP اللي هي الأدينين pocket، أو وين محل بيرتبط الأدينين الدرج بيرتبط. Besides طبعا other pockets that are not occupied usually by ATP. Okay. Now لاحظوا هون أنا عندي in this loop the aspartic acid of the DFG motif وكأنها pointing towards the binding site أو towards the active site. Okay, that's characteristic of an active conformation. That conformation fits it perfectly what we call or what we consider type 1. Type 2, those inhibitors, they actually favors the inactive conformation, where the DFG motif, as you can see in here, aspartic acid وكأنها pointing away from the active site. Okay. Type 3 and type 4 احنا مش رح نحكي عنهم بس انا حبيت افرجيكم اياهم. بتايب 3 برضو كمان let's say they bind to the hydrophobic pocket وممكن الالوستريك pocket اللي بيحتلوهم type 1 و type 2 بس none of the parts او خلينا نحكي structural scaffold اللي في type 3 بيحتل الادينين pocket فعشان هيك they consider it a type 3. Okay, يعني it does not form hydrogen bond interactions مع الhinge region. 4, it occupies a completely different position بالkinase, an allosteric site بالkinase. It does not approach neither الادينين pocket أو الhydrophobic pocket أو الallosteric pocket اللي بتكون قريبة من ATP binding pockets. Okay, as I've mentioned رح نحكي فقط عن type 1 و type 2. Here are two crystal structures of type 1 and type 2 inhibitors bound to kinases. Again, the N loop, the C loop, that's the C helix. This is the the ATP binding site. The one in blue is the glycine rich loop. This is the, let's say, loop that has the DFG motif. يعني هون مش كثير شكلها واضحة بس إنه as you can see the aspartic acid as if it's pointing towards the binding site. اوكي. هلا في عندي هنا يعني لو اخذناهم مثلا بالترتيب انه this is the adenine pocket, that's the hydrophobic pocket. اوكي that's the hydrophobic pocket. بيكون في عندي على شمال الهيدروفوبيك pocket what we call an allosteric site, which is, let's say, almost around here. Now that allosteric site لما تكون عندي ال DFG in ولا out in, الفينيلألانين بتكون مسكرة almost هاي الallosteric site. Okay. Now if you go to the type 2 inhibitors which favors an inactive conformation, لاحظوا إنه أنا عندي aspartic acid in here, it's pointing out. It's a DFG out. لاحظوا كيف بتكون الفينيلألانين وكأنه لما تحركت عندي الاسبارتيك يعني imagine them إنه this is the aspartic acid and that's the phenylalanine. لما صارت عندي الاسبارتيك acid pointing out, الفينيلألانين اتحركت والallosteric site in here is now open. Okay, for that allosteric part in type 2 inhibitor to occupy and bind in. All right. Size wise برأيكم مين أكبر؟ type 1 ولا type 2؟ type 2. Okay. بس مش دائما هي يعني مش دائما تاخدوها إنه هاي this is the base for distinguishing them. يعني الأساس إنه أنا أميز مين بيرتبط عندي في البايندينج ريجن أو في الأدينين باكيت وعلى أساسها بحدد الرينجز اللي مرتبطة فيها هل ممكن توصل للألوستريك سايت. If so, then it would be definitely a type 2. Okay. هلا بالليترشر هلا صار في let's say more thorough classification في type 1 and a half type, وبعض البيبرز بتصنفهم A and B. احنا مش رح ندخل فيهم، احنا بس رح نركز على type 1 و type 2 كيف structure اللي تميزوا بيناتهم. Let's take a first say example which is gefitinib. Okay. Now this kinase inhibitor لو سألتكم which part of this drug will form hydrogen bond interactions مع الهنج region أو in other words رح يرتبط في الأدينين pocket. أي وحدة؟ يعني لو مثلا رقمتهم 1 2 3 4. Yeah, this part in here is the one that will occupy الأدينين. Okay. طب برأيكم this one in here أكيد ما بترتبط بالهنج region بس do you think it will reach to the hydrophobic pocket? Yes. This one will occupy the hydrophobic pocket. Okay. هل بعتبر هاي الجروب hydrophobic in nature؟ بنزين عليها فلورين كلورين. احنا generally نعرف انه الهالوجينز usually they increase the lipophilicity of a drug, so I would consider this one a hydrophobic group. Okay. Now look at this crystal structure وكأني أنا بتطلع from the side. لاحظوا إنه أنا عندي this is the ring that is occupying this one in here, occupying الأدينين pocket, forming interactions مع الهنج region. Okay. This ring that has a chlorine and fluorine is reaching down to the hydrophobic pocket. إن شاء الله الأسبوع الجاي بفرجيكم let's say more elaborate crystal structures بتكون الصورة أوضح. Okay. Here's the let's say DFG motif. هلا لأنه من الزاوية هون عم بطلع بتبين وكأنه الاسبارتيك acid away بس actually if you would rotate yourself and look from here راح تبين الجلوتاميك اسف الاسبارتيك acid وكأنها pointing towards let's say الأدينين binding pocket. Its conformation is actually a DFGN. طيب what about الإيرلوتين؟ Yep, that's the adenine binding pocket, and this one will occupy the hydrophobic pocket. Here's a crystal structure for this drug. Again, as if you are looking, let's say, from the side. This ring in here, similar to gefitinib, is occupying the adenine pocket, forming interactions with the backbone of the hinge region, and this acetylene phenyl group is occupying the hydrophobic pocket. هلا those which are designated as solvent region بنحكي عنهم later on بس نحكي عن the design of gefitinib and why they are called let's say, why they were added to begin with to the drug and why they are, let's say, called a solvent region. Okay? What about this one? برأيكم, which part would probably bind to the adenine pocket? خليني أرقم لكم إياهم again. 1, 2, 3, 4, 5. Exactly, 1 and 2. This part in here is the one that will occupy the adenine pocket. Okay, الرينج اللي بتكون attached عليها الهيدروفوبيك, it's actually hydrophobic in nature, and this one can reach allosteric. So it's a type 2. All right. Here's a crystal structure. وكأني أنا بتطلع to the front of the enzyme. Okay. That's the N loop. That's the C helix. Okay. Now, here's the part that occupying the adenine pocket forming hydrogen bond interactions مع الhinge region. Okay. Here's the methyl phenyl that is occupying the hydrophobic pocket, and this one in here is occupying the allosteric pocket. This is the aspartic acid of the DFG. لاحظوا كيف بتكون بعيدة عن البايندينج. It's pointing away from the binding site. والفينيل ألانين يعني سوري إنها مش مبينة، بس الفينيل ألانين يعني بالactive conformation بتكون هون مكانها. blocking the allosteric site. Okay. When in the inactive conformation بتغير محلها، يعني بهالكريستال ستراكشر مش موضحينها بس بتكون let's say here in that position. It's not shown in here. يعني away وكأنها فتحت المجال for this part of the drug to bind in the allosteric site. Okay. What would you consider the nature of the amino acids in the allosteric site? Both actually. Some may be hydrophobic, forming interactions مع الفينيل, or some are actually, yeah, such as for instance this histidine forming interactions مع that tertiary amine اللي ممكن يكون let's say protonated at physiological pH. Okay. Here's another, let's say, a crystal structure of imatinib with the whole enzyme. If you zoom in, okay, لاحظوا what is shown in green, that's the hinge region and that's the DFG motif. يعني لاحظوا كيف عندي الفينيل ألانين بالactive conformation بيكون محلها هون. لاحظوا كيف when in the inactive conformation it rotates تبعد عن let's say the opening of the allosteric site. Lapatinib, type 1 or type 2? شو رأيكم؟ طيب وين الرينج اللي ممكن ترتبط بالأدينين باكيت برأيكم؟ رق ماشي 1 2 3 4 5. Yeah, this ring system in here is the one that will occupy الأدينين. Okay, what about this one? Hydrophobic. Okay, well this one can reach all the way to the allosteric. Okay. لا. The crystal structure of this one shows إنه الأدينين actually this one is what we consider an allosteric site. Yes. هلا if you would rotate it يعني ممكن هاي توصل ممكن توصل للهيدروفوبيك باكيت وممكن هاي للألوستريك بس بتصير the rest of the drug protruding outside of the binding site. If you would look at it, it's actually very hydrophobic. رح يكون في عندي let's say مجال كبير إنه يصير في عندي what we call a solvation penalty. يعني حتى لو تخيلت إنه الفيوران بتوسع وبتفوت بالهيدروفوبيك باكيت والسلفون بفوت بالألوستريك سايت بس the rest of the drug actually رح يكون outside repulsing with water. This would not be a favored interaction. A more favored interaction, those two phenyl rings will fit into the hydrophobic of those, let's say, two phenyls بالhydrophobic بالallosteric. Okay. طيب, what do you think the interactions that this ring, the fluorophenyl اللي ممكن تعملها مع الallosteric? Yeah. So in other words, الامينو acids اللي موجودة بالallosteric site are diverse. They are not conserved. يعني in some for some kinase inhibitors they can be let's say ionic أو ionized أو ionizable. ممكن يكونوا very hydrophobic let's say highly polar. It depends. فيعني الالوستريك site provides us with a room for diversity أو a room for more selectivity to kinases. السورافينب و this one by the way بالsolvent region again رح نحكي عنهم later. السورافينب type 1. طيب وين الرينج أو خلينا نحكي the ring or the ring system اللي ممكن يرتبط في الأدينين pocket اللي بالنص. Yeah, this one in here. I would expect this is the one that will bind in the adenine pocket. Okay, what is connected to it can reach to the hydrophobic, and this one can go into the allosteric. Okay, يعني وكأنهم rings متصلين في بعض. Ring systems مش fused, linked together. Okay, هذا بالضبط. It's type 2. It is type 2. Okay. طيب, we will stop here. إن شاء الله المحاضرة الجاية في مجال I will show you let's say the crystal structures of those drugs على أساس إنه نشوف in more detail how they bind in the binding site والallosteric site and so on. وراح نكمل إن شاء الله specific let's say kinases as being targets for other drugs. وبرضه راح نحكي عن the design of gefitinib وimitinib. Okay. Any questions so far?